Hello, I'm Luca Maccioni and I'm a postdoctoral fellow in the Laboratory of Liver Diseases in the National Institute on Alcohol Abuse and Alcoholism under the supervision of Dr. Bingao. I'm going to show you our recent findings published in GATT. Alcohol-associated liver disease is a range of health problems that affect your liver when you drink too much alcohol. This disease can start out mild but get worse over time, leading to serious liver damage. Now, your liver doesn't get hurt just because alcohol touches it directly. It also gets affected through something called organ crosstalk, which means different parts of your body are talking to each other, and in this case, mostly your gut and your liver. This is known as the gut-liver axis. Recent studies focus their attention on paracellular intestinal permeability, dysbiosis, and effective secretion of antimicrobial molecules. However, the intestine is home of a large variety of immune cells which are particularly affected by alcohol. In this regard, we have recently demonstrated apoptosis of duodenal CD8 T cells in human ALD, while in the liver, a subset of patients show very high level of CD8 T cells. However, their mechanistic role in ALD remain unknown. In this study published in GATT, our mechanistic and translational approaches with mouse and human samples showed that in the duodenum, the home of a large number of CD8 T cells, extremely high level of ethanol after alcohol consumption promote loss of duodenal CD8 T cells, a process linked with increased microbial translocation. Our next analysis uncovered a unique downregulation of the pro-survival protein BCL2 in duodenal CD8 T cells, which is crucial for memory T cell survival. Excited by these results, we therefore determined the mechanistic role of BCL2 by generating CD8 BCL2 transgenic mice with BCL2 overexpression specifically in CD8 T cells. Our results showed that compared to wild-type mice, CD8 BCL2 mice had lower levels of serum LPS and ALT, revealing a key role of duodenal CD8 T cell reduction in the pathogenesis of alcohol-induced gut barrier disruption and liver disease. In addition, we are investigating the role of intrahepatic CD8 T cells in ALD. And as you all know, in healthy condition, the main population of immune cells in the liver are resident and Kupffer cells, and there are very few neutrophils and CD8 T cells. However, in ALD, we can find reduced number of Kupffer cells, but high number of infiltrating macrophages, neutrophils, and also CD8 T cells. But especially the role of CD8 T cells is unknown. We wanted to understand what CD8 T cells inside the liver are doing. We were lucky to get access to liver transplant sample at Johns Hopkins University, and we used a special technique called single-cell RNA sequencing to study liver cells. Our analysis showed that in patients with severe alcohol-associated hepatitis, liver CD8 T cells had high levels of survival proteins like BCL2, which help the cells stay alive. This was the opposite of what we saw in the gut. These liver cells also had high levels of inactivation markers and lower levels of inflammation-related genes, suggesting that activated CD8 T cells in the liver are not causing damage. They are actually helping protect the liver in people with alcohol-associated liver disease. We finally wanted to see what CD8 T cells do in alcohol-associated liver disease. And we tested this in two groups of mice, normal mice and mice that don't have CD8 T cells. At first, we gave both groups alcohol and we saw no big difference in liver damage. This might be because the liver normally has very few CD8 T cells and that the disease model we used was mild. Next, we used a stronger model that included alcohol and a molecule called 6L1, which causes more immune cells to enter into the liver, similar to what happens in humans. This made more CD8 T cells show up in the liver, especially after alcohol consumption. And when we tested this stronger model in mice without CD8 T cells, their livers got worse. They had more inflammation, oxidative stress, and troubles managing cell death. So even though CD8 T cells are part of the immune system, they actually protect the liver during alcohol-associated liver disease. In conclusion, our study suggests that promotion of duodenal CD8 T cell survival and functional activation of intrahepatic CD8 T cells could serve as a potential therapeutic target to treat or prevent alcohol-associated liver disease.